Hi, everyone. Today, I'd like to introduce Dr. Christoph Guger. He is the founder and CEO of g.tec Medical Engineering, leading company in the brain-computer interface and biosignal processing space. Dr. Guger has studied biomedical engineering at Johns Hopkins University and received his PhD through the Department of Medical Informatics at University of Technology, Graz. He developed the first real-time BCI system with continuous feedback. Additionally, he developed real-time analysis techniques with common spatial patterns for oscillatory BCIs. So, Dr. Guger, can you tell us a little bit more about your story? I'd love to hear about your background and your journey so far.
Hello, and thank you very much for the invitation. So I studied at Johns Hopkins University as a visiting student many years ago. And at Johns Hopkins University, they told me about a professor with the name Gerd Pfurtschel, who worked at my home university, University of Technology in Graz, on brain-computer interfacing. And of course, I didn't know him because the university is very big. And normally you have to go far away so that you figure out what's called at home. And after Baltimore, I returned to Austria, and then I applied for a master thesis in Gerd Pfurtscheler's lab. And so I started to work on brain-computer interfacing. This was 1996, so pretty long ago. And just a year later, I started my PhD. And at the end of my PhD, people were already asking if I would also sell my brain-computer interface that I developed during the dissertation. And so I established g.tec Medical Engineering, and actually the first brain-computer interfacing system was sold to Oxford University in England, and the second one went to South Korea, to a big research institute in South Korea, and actually I forgot the third one. So g.tec is already 25 years old. This is my company, who is nowadays producing and designing different brain-computer interface applications.
Sounds like it's been an interesting and exciting journey so far. And so I'm definitely looking forward to diving into about g.tec and some of the specifics about what you've been working on. But before that, I wanted to ask what originally sparked your interest in neuroscience or neurotechnology?
So at this time, I made the decision between building wind power plants and going into the brain-computer interface domain. And at the end, wind power plants were too political for me because you always need, the allowance to build it somewhere. So I didn't want to have that. And in brain-computer interfacing and neuroscience, it's much more flexible. So demand and need is dictating what you can build. You don't have to ask any politicians if you're allowed to build it somewhere or use it somewhere. And neuroscience is anyhow fascinating, and especially to extract in real-time information of the brain. 25 years ago, there was not so much going on about the brain and EEG. So not so many people worked on that, but technology developed with high speed, and nowadays we can just do much more in this domain, which is of course fascinating.
I'm glad you ended up choosing this path. In my opinion, the field is lucky to have you.
Thank you.
And so now that we know a little bit more about your story, can you give us some background on the timeline of g.tec Medical Engineering? So you mentioned you started them just coming out of your PhD dissertation. Now the company's been around for, I believe you mentioned, about 25 or more years. And so what's happened between then and now?
So when I started my PhD, people were working on neural networks for brain-computer interfaces, and basically they acquired EEG data over many days, and then they were feeding, these hours of data into a neural network. So they had different implementations and different algorithms, and they trained it for many, many weeks on the data because computers were not so fast at this time. And my step in the PhD was that I used only 5 minutes of training data. So I put the EEG data into the calibration phase, and I could more or less immediately control a cursor. So this was a big step forward because instead of weeks of calibration, it was reduced to a few minutes. Nevertheless, to get high accuracies with a multi-imagery based brain-computer interface, we needed a couple of hours of real-time feedback training to achieve it. And you mentioned before in common spatial patterns was one algorithm that we developed during my PhD. And it was fascinating because the first experiment ever that reached 100% classification accuracy happened with common spatial patterns. So this was a big step many years ago, because then the feedback was much more useful and people learned much faster to control such a brain-computer interface. At this time, we were also working with passive EEG electrodes. This means you had to abrade the skin and clean the skin to mount EEG electrodes. So for 64 channels, it more or less took 1 hour to prepare somebody, and you can imagine the people were already tired before the experiment started. And then with g.tec, we started to develop active electrodes. This means the amplifier is sitting directly in the EEG electrode. And this changed everything because we could reduce the preparation time for 64 channels to 5 minutes. So this means in minute 6, you could already start your experiment. So people were fresh. And overall, we got about 10% better classification accuracy in all our brain-computer interface experiments because of that. And a few years later, we released Intendix. This was the first P300 spelling device that people could use for communication. So it was developed for locked-in patients and ALS patients who couldn't move anymore. And special about Intendix was that we needed only 3 minutes of calibration data, and then already somebody could spell without making mistakes. For that, we used the so-called face speller. So we overlaid these black and white characters with famous faces. This was pretty innovative about 20 years ago. So we had Brad Pitt over the A, for example. And when the patient saw the Brad Pitt flashing, then his brain tried to identify the person, the face. This produced a really big P300 response. So it was much easier for the brain-computer interface to detect it. And with this setup, we actually reached 100% accuracy for everybody. So it was really stable. But nevertheless, we needed a couple of minutes of calibration data. Nowadays, we are using CVAPs, so code-based visual evoked potentials. With this technology, we can reduce the calibration time to 20 seconds. And it works also in Unity, so you can work with head-mounted displays in VR, in AR, to give highly immersive feedback. And the reaction time is also much better. So we need on average 2.5 seconds for making a decision for selecting a new item in Unity, for example. So this shows that the accuracy and the calibration time improved a lot. And also we use the technology nowadays for many different applications, which just didn't exist 25 years ago, where we basically move the cursor to the left and to the right-hand side.
It sounds like you've worked on a lot of cool projects and brought a lot of efficiencies in terms of timing, as well as in terms of classification accuracy to the space. And some of those stats you were giving me and how big of a step change that these algorithms that you were working on caused were just really exciting to hear. And so now I wanted to dive a little bit more into a specific product. I'd like to hear more about the RecoveriX. I was reading about it on the g.tec website, and it's my understanding that it's an EEG-based BCI neurorehabilitation system, and it looks like it was designed primarily for stroke patients, and then it included a VR and functional electrical stimulation component. Could you just share a little bit more detail about that system and about how all these components work together and aid in the recovery and rehabilitation?
Yeah, g.tec started with research products, so we have many elements which are used for different non-invasive and invasive research applications. And a few years ago, we started also with medical applications, and RecoveriX is one of these medical products that we are producing. So the purpose is to treat stroke patients and patients with multiple sclerosis. So they have some motor deficits because of the illness, so they cannot move the hands or legs correctly, and we wanted to treat that. So for RecoveriX, we use a wireless EEG headset with 16 electrodes over the sensory motor cortex, so that we can pick up hand movement imaginations or foot movement imagination. And in front of the patient, we have a computer screen where the patient can see a little avatar. The avatar is moving either the left hand, the right hand, or the foot. And this is the instruction for the patient to imagine the same type of movement. So let's say The avatar instructs the patient to imagine a left-hand movement. Then with the brain-computer interface, we can pick it up in real time that the patient is now doing this. And then we trigger a functional electrical stimulation of the left hand so that the hand is really moving. So this is a lot of motivation for a stroke patient, especially if they were paralyzed for a couple of years. So suddenly the hand moves again as soon as they imagine the hand movement. This is exactly what we want to achieve. And in one training session, we are treating left hand versus right hand movement, for example, for the upper limb treatment protocol, and they have to imagine it 240 times. And they are coming back for 25 treatment sessions. And altogether, they have to imagine the hand movement 6,000 times. And this is so often like a baby needs to learn to walk. This is producing neuroplasticity. This means healthy neurons are taking over the task to control the hand or the leg, and damaged neurons are replaced by this training. And with EEG biomarkers, we can very nicely see that this neuroplasticity is happening. And it's also very nice that we are running 18 different functional tests just to objectively show the motor improvements. And in these patients, we get, for example, better fine motoric skills. This is tested with a 9-hole peg test where you have to stick 9 little sticks into certain positions with the healthy and the paretic hand. Then we have the box and block test for cross motoric skills. So they have to move little blocks from one box to another one. And for the leg treatment, they have to walk, for example, 10 meters. We are measuring the time. Then they have to walk around for 6 minutes. We are measuring the time. They have to stand up, move along a circle, and sit down again. We are measuring the time. And with these tests, we can very objectively show the improvements. And basically, we get better motoric skills, but also reduced spasticity. And we have also a lot of side effects, but they are all good. For example, the temperature control of the paretic limb gets normal again. Concentration performance improves. Memory improves. Pain is reduced. In Finland, for example, RecoveriX is already used for many years for chronic pain. So if medication doesn't help anymore, they do hand movement or leg movement training with RecoveriX. And my favorite side effect is actually that we get better speech. The explanation is that we are activating the sensory motor cortex. And if you're looking at the language network, then you need, of course, the auditory cortex to hear language. Then you need Wernicke's area to understand what somebody is saying. Then you need Broca's area to formulate the answer. And finally, you need the motor mouth region moving the lips and the tongue to formulate the word. And this is the region that we activate. And so we have a positive effect on the whole language network. And that's the reason why people speak better after they trained with RecoveriX. The second clinical study was actually for multiple sclerosis. And we also figured out that these patients get much better. So it's a game changer for MS patients because normally they get medication to reduce a little bit the speed of the decline. So all the neurologists are telling MS patients it will get worse and worse. You have to take medicine against it. And with RecoveriX, we can actually reverse it. So if they train with RecoveriX, they get better again. And this is, of course, very important for these patients because they're much younger than stroke patients. So MS on average occurs when people are around 34 years old. So they are pretty young. And of course, it's very helpful to have a technology that helps them.
I was really excited to hear about all the positive side effects that came with this system that maybe it wasn't specifically targeting, but things that it ended up helping out as well. And I'm also glad that it's a non-invasive system because I think it's helpful for getting it into more hands and getting more patients treated with it that way. So I did have a quick follow-up question. You mentioned that there was some sort of EEG biomarker that you guys have been picking up related to the plasticity or the retraining of certain neurons in the motor area. I was wondering if you could speak a little bit more about that.
So we are having 16 electrodes over the sensory motor cortex, so left-hand region, right-hand region, and foot region. And we are instructing the patient at a certain time point to imagine the hand movement. And then we can see that the sensory motor cortex over the specific region gets active. This biomarker is called event-related desynchronization. Actually, Gerd Purcell, my PhD supervisor, came up with this principle. And it basically means as long as you're doing the hand movement imagination over the sensory motor cortex, the alpha and beta oscillations are dropping down, so they get smaller. This is the biomarker that we use for pinpointing neuroplasticity.
You basically notice like over time that biomarker is showing up more often.
Yeah, the EEG gets stronger. And what we typically see at the beginning that people need about 3 seconds to activate the sensory motor cortex, and then we find a difference between left and right. And after a couple of training sessions, the sensory motor cortex gets faster and faster. And after 25 training sessions, very often the sensory motor cortex needs only like 200 milliseconds to get activated and to show a difference between left and right. And also the difference between left and right hand movement imaginations gets bigger and bigger with training. And this is exactly what we want to achieve. And this is also the neuroplasticity that we want to produce.
Yeah, absolutely. Those results sound incredible. And so taking a quick pivot to what might be more of a research tool, I had a few questions about that g.tec BCI system. It seems like it could be useful for a variety of applications, and I was reading about the system. I think it was called g.bcisys, and something that stood out to me was the rapid prototyping functionality, and it mentioned that development times could be shortened from several months to just a couple of days, and it seems like that really goes with some of the themes of efficiency that we were talking about earlier. And so my question was, who would be the ideal customer for this kind of system? Would it be an EEG BCI research lab? Would it be like clinicians, or both?
Both, and also industry. So we have a lot of industrial customers. So the idea of the rapid prototyping system is that you take one of our hardware components. We have different biosignal amplifiers. So they range, for example, from 8 up to 1024 EEG channels or ECOG channels. And then you can take either EEG electrodes for surface recording, non-invasive recordings, but you can also implant ECOG grids. They're made of platinum directly on the cortex. For that, you need a neurosurgeon. And then you can do also invasive recordings. And you can also use depth electrodes. And you can stick it, for example, into the thalamus. Then you can also record from deep structure. And then you can also select some of our stimulators. So we have electrical stimulators that can be used in the cortex. So you can stimulate on the surface of the brain, but also into deeper layers. Again, like the thalamus, STN, in Parkinson patients, for example. With our software, the g.tec suite, you can combine neuromodulation or closed loop experiments. This means you can sense from a specific region of the brain, you can extract your features or biomarkers, and based on the results, you can stimulate into different regions of the brain, or you can also stimulate the periphery. So you can place electrodes on different muscles, and when you provide functional electrical stimulation, then the hands or the legs are moving. And with these closed loop possibilities, you can do many, many different applications. And all the experiments become more efficient and also more exciting. And with the rapid prototyping environment for a new experiment, normally we just need a few hours, because the data acquisition, the feature extraction, the classification, the control of the stimulators, that's all there. And you are basically just adapting it to your experiment. And we're also helping clients for doing that, and they use it in many different domains, for example, with epilepsy patients, with tumor patients, for different neuroscience experiments, and also in the non-invasive domain, for example, for stroke and multiple sclerosis patients, like we are doing it with RecoveriX. But you can also use it for flight control if you work with the Air Force, for example, to extract information biomarkers of pilots or flight controllers and so on.
Wow, that seems incredibly customizable. And as you mentioned, it seems like it can be used for quite a variety of applications. That's a pretty awesome system there. So next, I'd like to talk a little bit about a recent research publication. You were listed as a co-author on a publication titled Recording Physiological and Pathological Cortical Activity and Exogenous Electric Fields using graphene microtransistor arrays in vitro. And so the publication goes on to talk about the testing of graphene-based solution-gated field effect transistors. The study notes this is still an emerging technology, but it does accomplish a pretty in-depth in vitro characterization of these transistors for clinical brain recordings in human tissue. But mentioned one of the key potential benefits is that these would allow for the quantification of the full band of the brain signal. And so that would include flow wave DC potentials that are not captured in most of the common systems of today. And so I was just wondering if you could talk a little bit more about the potential impact of that work and maybe what some of the next steps towards optimizing that technology or moving it to the clinic towards the clinical domain or research domain would be. And then also, if there's any plans for g.tec to include that technology in any of their projects or in the future.
So this project is running under the Graphene European Union flagship program. So it's a 10 years project where many different universities and also companies are working together. Our part in the project was to design the biosignal amplifiers. So we used our 256 channel biosignal amplifier g.biomarker. This is a nice tool because we can sample very high and we have also a very high resolution with 24-bit and a very low noise floor, which is achieved by a dramatically high oversampling and anti-aliasing filter. So the unit has 4 processors inside to achieve all these things. And the nice property about graphene is that it's cheap to produce and it's very thin and it's invisible. And you can also put electronic circuits into the graphene electrode grid. And University of Barcelona designed such an electrode grid, and we interfaced it with our biosignal amplification toolbox. And this was tested at University College London and at EEG Biomarker in Barcelona, so with different animals. And a big advantage is that you can produce very tiny electrodes, so you can also record from single cells. And this is typically done with electrodes made of other metals. They have the problem that they polarize. This means there's a little DC voltage building. And this makes it very difficult to record slow waves or DC potential. And for graphene electrodes, this is not the case. So the polarization more or less doesn't exist. For that reason, you can also record very nicely DC potentials. One of the possible applications is migraine or cortical spreading depression. So this wave starts normally over the visual cortex. So people with migraine know it, the eyes start to flicker. And then the wave, the cortical spreading depression, flows over the whole brain, and then you get a headache and all this stuff. And with such transistors or such graphene arrays, you can study such effects very nicely. And this would be very difficult with other materials which are produced of standard materials.
Those migraines and those other conditions that you mentioned, those are highly related to those DC potential?
Cortical spreading depression is basically analyzing DC potentials. So this is how they are decoded. So if you look at oscillations or spikes, you are not really seeing this information. But DC potentials are not used for so many different applications because it's so difficult to measure them. So most of the brain-computer interfacing is done with oscillations, alpha, beta, gamma region, or with single units. So people are looking at neuron spikes.
And that's exciting that your company has already been able to develop an amplifier that interfaces effectively with this new technology that they're studying. I wanted to make a pivot once more and talk about how you and your company do a lot to drive community in the neurotechnology space. That's with initiatives like the BCI Awards, the Neurotechnology Spring School, and the Brain.io Hackathon. And so I was just wondering, first off, what was the inspiration for these initiatives? How has the journey with growing them over time been?
So we can start with the Brain-Computer Interface Award. This started more than 10 years ago. When I started with brain-computer interfaces, there were basically 3 labs all over the world active in this domain. Now we have many hundreds of them. And, every single year, more and more labs started to produce nice applications. That's the reason why we wanted to create an award to make the community aware of all the cool stuff that's developed. And each year we are nominating 12 projects of all the submissions, and then we have a jury, and they look at these nominations and select the first, the second, and the third place winner. And then we have also a BCI award ceremony where all the nominees can present their work, and then also the winners are announced. And this is very nice because it's actually a huge event from many different countries and labs. People are participating and they can just showcase what they develop. They get also awards and some prize cash money, which is also nice, and they can list it in their CV. And if I'm nowadays getting, for example, an application, if somebody wants to work with g.tec and I see that they were nominated or even winning the BCI award, I know that they have a very good idea of what they are doing. And if I look just at the budget, that the nominated projects need, then you can also see how much the technology is actually pushed. So 10 years ago, it was a very small amount that you needed to realize all these research projects. And nowadays we reached several $100 million, which are invested just in the 12 nominated projects. So this shows how much the technology is skyrocketing. And also, if you look at the number of different applications, 10 years ago, many people did offline analysis, they classified EEG data. Nowadays, almost everybody is doing real-time experiments. So it's fascinating how much the technology progressed. And this triggered also the idea for our hackathons, the Brain.io hackathon series. So these are events which are 2 days long. People are meeting over the weekend. We put them into teams of 5 people normally, and then they have to work together for 24 hours to come up with new and innovative applications. And we have also an international jury. So at the end of the hackathon, they have to present the project. The jury scores the different projects. They can also get awards and cash money, but more importantly is it's very nice for collaboration. So they learn to know each other, they maybe meet their future PhD or master degree supervisor, or they find their intern position. In the last hackathon, we had 113 countries working together in a lot of teams, and every year it's getting better and better. I also participated many years ago in a hackathon, and after 24 hours they didn't reach much. But nowadays, with current BCI technology, they get within 24 hours real-time applications running, and it's just fascinating to see what they can achieve in such a short time frame. And also the projects are getting more and more innovative and fascinating, so I invite everybody to participate. And the next one is actually embedded in our BCI and Neurotechnology Spring School. So this one is happening in April and May again. It's 10 days long, and we invited 140 scientists and people also from companies to speak about their BCI and Neurotech project. So we have all the top universities on board as speakers, and the hackathon is actually embedded on the weekend, and we expect that it will be huge again. Last year, 2023, we had almost 16,000 people participating from 113 countries worldwide. And I guess this year it's getting bigger again. But it's just a very nice kickstart if somebody wants to go into BCI and neurotechnology. So after 10 days, you have basically seen what's going on around the world, because we have such good speakers. So we handpicked just the best speakers that we met in the last year and who are really doing innovative projects, and then you can just learn it very, very quickly within 10 days. Person Matt, for example, will explain how he records EEG data in dolphins. Not many people are able to do that at all, and Matt will show us how this is actually doable.
That's a great way to dive in and get a good introduction to our technology field. And even for people that are familiar with the field, just hearing from all these speakers just seems like a very interesting and fascinating experience. Another thing that resonated with me was just how much people can get done in a short 24-hour hackathon these days. It really is impressive and fascinating, just the speed that people are able to get something up and running. What is next for g.tec Medical Engineering? Are there any new products or future initiatives on the horizon that you wanted to talk about?
Yeah, we are getting more and more into neuromodulation, and we are building more closed loop experiments. So this is very interesting for the invasive domain with epilepsy and tumor patients. So if you can map the eloquent cortex of patients more precisely, then the neurosurgery outcome is, for example, much better. So if the doctor understands better where important centers are located, then they can spare it and they don't resect these regions. And on the other hand side, if they know these regions exactly, then they can resect more of the tumor. This also means people are living longer after the tumor resection. And these are just very important points. And then with neuromodulation experiments, every year there are new applications coming up. Depression is, for example, a big topic nowadays. With invasive brain stimulation, by stimulating different brain regions, you can reduce depression. So it's already done for many years in Parkinson patients where the thalamus is stimulated, and then you can reduce the tremor. But this technology can also be used for many, many other illnesses, and it's just spreading out and explored by many, many research centers. And very interesting is also to use brain-computer interfacing in daily applications for end users. We are going, for example, into the gaming environment with Unity. So our new unicorn interface is just streaming the data directly in Unity, and we have also toolboxes which are taking care of all the feature extraction and classification. So basically you can just put controllable elements into your computer game, and you don't have to worry about the EEG signal or the feature extraction at all. It's all handled. So it's basically a Unity asset that people can just use. This will make it super easy for developers to utilize BCI technology. This is also very interesting, of course, for neuroscience experiments, because you can just use all the tools, these head-mounted displays, for example, and come up with new applications.
Sounds like innovative and exciting times ahead. Based on the projects that your company is working on, it sounds like they're going to be involved in even more neuroscience research, helping the lives of even more patients as you guys expand the gaming industry and these various other clinical applications that you mentioned. Next, I wanted to take another pivot and ask about advice that you would give to students or young professionals aiming or hoping to work in the field of neurotech. First off, some good advice might be for them to attend some of these initiatives put on by g.tec, for example, the hackathon or the Neurotechnology spring school. But I was wondering if you had anything additional that might be useful for those trying to break into the field.
Yeah, BCI technology grew tremendously. So there are so many different domains where you can work nowadays. So you have to identify what you really like. So maybe you want to work with neurorehabilitation with stroke patients or multiple sclerosis, or you want to go into neurosurgery and help neurosurgeons to optimize procedures, or you just want to be in the gaming domain and use EEG signals. So at Spring School, every day has a certain topic, and then you get a very nice state-of-the-art overview of what people are doing in neuromodulation, in high gamma mapping, in cortical network mapping or whatever. So it could help you to identify the most fascinating topic for you. And then I suggest, of course, to go into another lab or to do an internship project or a master thesis, PhD thesis together with another lab or a company, so that you see exactly how this is done and so that you can learn how to do it, how to run the experiments, how to deal and acquire EEG data with patients and so on. So there are a lot of different things to learn, and you must be embedded in a field with experts so that you can learn quickly, and then you are reaching a state of the art which is producing a lot of fun, and then it's also easy to get scientific publications or your degree much faster. But go to another lab and learn what they are doing, and then pick what you like.
Yeah, that's great advice there. You gotta find your niche, and you also got to find good mentors to learn from and labs working on things that interest you. So do you have any predictions for the future of neurotechnology over the long term, perhaps over the next 20 years or so?
Yeah, we will see much more neurotechnology even in the next years, but in 20 years it will be dramatically oft used. So RecoveriX is a very simple product at the moment, where we can already see these benefits. So up to now, people or neurologists are telling patients, if you have a stroke, there's some progression in the first year, but after that, you are stable and you don't get better anymore. And with RecoveriX, we have shown this is just not the case. Even 10, 20 or 30 years later on, people are getting better. They just have to participate in the training. And we established also a franchise system, so that therapists or businessmen can use the brain-computer interface in their own therapy center to treat patients. And this is just spreading out nowadays. So this is in addition to standard rehabilitation centers and hospitals. So we will see just in this domain many institutes or recovery centers around the world very soon. And in 20 years, there will be many offering this type of technology. And this is just one application. You can also do brain assessment of concussion and so many other topics. So we will see much more. And I mentioned before that RecoveriX is also used for chronic pain patients. So this also shows that we will have options to replace medicine, or at least to add some benefits on top of medicine. This is also very nice because if we don't have side effects, people live much better.
Sounds like lots of new indications and applications for neurotechnology on the horizon. It's really exciting and fascinating to learn that we're starting to see major plasticity even in these patients that had had a stroke 30 years ago. So that's great news and exciting news and really helpful for these patients. And so that was the final question that I had for you. Did the listeners contact you for any further discussions after the episode? And if so, who should reach out to you? How should they reach out to you? And then what topics should they reach out to you about?
So this can be a student who wants to do a bachelor or master thesis, or somebody who wants to do a PhD or a postdoc. So we have many researchers and interns in the company. So we have also different locations where people can join us in the US, in Canada, in Colombia, in Spain, in Austria, and in Hong Kong, and also in Japan. But this is more for engineers or therapists, neuroscientists. And of course, if you know somebody who suffered a stroke or who has multiple sclerosis or other neurological disease, then let them know about new neurotechnology. And maybe it's a good treatment options for them to get better again. And for the brain-computer interface and neurotechnology spring school, so invite your faculty, your friends who are interested about this topic. So it's free. Just register and then you can join the event via Zoom. So you can stay at home and just listen, or convince your teacher in school or your professor at the university to make it part of the curriculum. And then you can just participate, and you also get some credits from universities. So we're also providing 14 credits for the spring school, because it's 140 hours long. It's like a 1 semester course, more or less, or the courses that you take within 1 semester. And we have universities who are already accepting the spring school for their curriculum. So you get credits at local universities. So just inform your professors and maybe you'll get credits for it.
Yeah, that's awesome that the Neurotechnology Spring School is accepting credits, and yeah, that's a lot of learning really condensed into that 10 days. So I just wanted to say thanks again for coming on today. The conversation was really informative. I feel like I learned a lot from you, and I really appreciate you taking some time out of your day to come talk with me.
Thank you very much for the invitation. It was very nice.
Yeah, absolutely.